my name is Ashley. I'm a part of the MCB 540, and I'll be exploring the genetic factors in Alzheimer's disease using the UCSC Genome Browser. So an introduction to AD, it's a progressive neurodegenerative disorder that's a leading cause of dementia worldwide. The symptoms include memory loss, confusion, difficulty with language and problem solving, and ultimately leads to a loss of independence. The pathology in the brain is characterized by a buildup of abnormal proteins, these beta amyloid plaques that we'll talk about later, and also tau tangles. It causes nerve cell death and connection breakdown in the brain. So the purpose of this project is to explore the genomic regions associated with AD susceptibility using the UCSC Genome Browser. Some background: the strongest risk factor for AD is advanced age, with the second being family history. So these genetic factors are estimated to play a role in at least 80% of AD cases. We know that rare mutations in APP, PSEN1, and PSEN2 almost guarantee early onset of familial AD, and that represents about 5% of cases. We also see some common gene polymorphisms in the APOE gene variants, E4 and E2, which influence susceptibility for about 50% of common late onset AD. So these four genes, APP, PSEN1, PSEN2, and APOE, account for about 30 to 50% of AD inheritability. And so I'm really going to be focusing on these four genes in my project here, but it's also important to count that GWAS has identified 11 additional AD candidate genes that's possible to look into. So the hypothesis and goals is that these genetic variants within these susceptibility genes that I had mentioned before, when using the UCSC Genome Browser, will show some tissue-specific expression patterns, which will suggest involvement in these regulatory mechanisms in AD pathogenesis. I also expect to identify some genetic variants with potential functions on gene expression and protein function, which provides insight into the genetic basis of AD susceptibility. Then further down the line could be potential targets for therapeutic interventions. So the methods that I used, I accessed the UCSC Genome Browser and used the most recent genome assembly for humans, and I looked up these four genes that I had mentioned before: APOE, APP, PSEN1, and PSEN2. And then I looked at the different genomic tracks for AD. I looked at the different genetic variants, the gene annotations, the regulatory elements, and then I also explored different features such as the exons, introns, enhancers, and these genetic variants within these regions that could influence how that gene is functioning in the brain. And then from there, I looked at tissue-specific expression, and so I used those tracks on the genome browser, and I compared them across different brain tissues, and then also different relevant tissues in the body. And so the first is APOE. Just a little bit about APOE. It's a pretty important lipid. It's a pretty important protein in lipid metabolism, and it really shows the importance of in neurological health. It transports cholesterol and it clears it from the brain, and so it's pretty important for neurological function and maintenance. Of particular interest are the different alleles in the APOE gene. So the E4 is strongly associated with increased risk of developing AD, while the E2 allele seems to provide some protection against the disease. And so we look at the transcripts here. You can see that there are some short genetic variants that you can see along the exons here, as well as the allelic variant phenotypes that correspond with the exons that could be the different E4 and E2 alleles that are variants that show different functions, as well as we see different repeating elements. So here we see the signs, which are just different transposable elements. When we look at expression across all the different tissues, interestingly, the brain doesn't have the highest expression, but when we take into account the cholesterol functions, we actually see the highest expression in the adrenal and the liver tissues. APP is an amyloid precursor protein, and it's also a key player in AD. The protein's found in high amounts in the brain and is involved in various cellular processes, so you also see it throughout the entire body. It has an importance in the production of amyloid beta peptides, and these peptides are known for forming the plaques in the brain that we talked about earlier. While the exact function of APP is still trying to be found, it's believed to play a role in some neuronal growth, repair, and synaptic functions. Mutations in the APP gene can cause an overproduction or altered processing of these amyloid beta peptides and can contribute to the development of AD, and particularly in familial cases. So we know that APP has a strong inheritable factor. We see the transcripts here. A lot of what we see here are introns, but there's a lot of short genetic variants, as well as transposal elements, LTR, a lot of different repeating elements we see in this APP gene. When we look at expression, we see very variable expression all throughout the body, but we see the highest expression in the frontal cortex in the brain, and outside of the brain, we see the highest expression in the thyroid. Next, we looked at PSEN1. It's a protein that plays a pretty critical role in the development of AD, and it's a key component in the gamma secretase complex, which is responsible for the cleavage of these APP proteins and into the amyloid beta peptides. The amyloid beta peptides are known for their aggression into plaques. The mutations in the PSEN1 gene are strongly associated with early onset familial Alzheimer's disease, where symptoms usually appear before the age of 65. Beyond its role in APP processing, PSEN1 is involved in various cellular functions, including calcium homeostasis, synaptic plasticity, and other cellular functions. Here we see the different transcripts for PSEN1, and it also has a lot of different short genetic variants and repeating elements. So we see here more transposable elements, retrotransposons, and different long terminal repeats. Here we see that the spinal cord has the highest expression across all tissues, which is rather interesting. Lastly, we looked at PSEN2. It has a lot of the same functions as the previous one, PSEN1. You see here the different transcripts, the short genetic variants, as well as the repeating elements. So the repeating elements mainly have transposable elements, and then there's a lot of different short genetic variants that you see across this gene. And then as well, we see a lot of PSEN2, you see a lot more variants throughout the body of gene expression, but the highest expression patterns in the brain you see in the cerebral hemisphere and the cerebellum. So overall, we looked at the genomic regions containing that are known to be susceptibility genes. That's APOE, APP, PSEN1, and PSEN2, and we looked at tissue-specific expression, and we saw different expression patterns across the brain tissues that some were quite surprising and some we could link together, and this emphasizes the importance of tissue-specific regulation in AD pathogenesis. Why this is important in the field of genomics and in science and Alzheimer's disease research, it's crucial for diagnosis. So understanding the genetics facilitates the development of targeted diagnostic tools. It's also important for treatment. It identifies some novel therapeutic targets for drug development. It's also crucial for prevention. So this informs different strategies for disease prevention and risk reduction. So in conclusion, when we're able to leverage these tools used by, for example, the UCSD Genome Browser, but there's a lot of different other tools available, it also offers some valuable insights into AD genetics when we didn't have to generate the data ourselves, which is amazing. We saw some tissue-specific expression analysis, and it provided some additional context for understanding AD susceptibility. And understanding these genetics is really important for advancing our knowledge and developing some effective interventions to combat disease. Here are my references. Thank you so much.